We'll be covering transcription and translation, which is the process that DNA is turned into proteins in the body. And in this video, we'll also talk about DNA, mRNA, rRNA, and tRNA. So transcription is basically DNA to mRNA. And this step involves the protein DNA RNA polymerase. And RNA polymerase is the main enzyme, like protein, involved in transcription. It reads one of the DNA strands and match complementary nucleotide bases to make an mRNA, to make this. And something to notice is that instead of thymine, A to T, we have A to U, so thymine is replaced by uracil. And so this mRNA will later be translated to protein in a polypeptide, but transcription is DNA to mRNA. And so the difference between uracil and thymine is just they lack this. They're pretty much the same thing. And so what is mRNA? mRNA is the product of complementary nitrogenous bases with an uracil instead of thymine that we got from the DNA. And mRNA is called messenger RNA because it's supposed to carry this transcript, this message to the ribosomes to produce protein. And mRNA is separate, like it's in chunks called codons in three bases. Three bases will be translated into amino acid, which will build up to proteins. And the difference between mRNA, rRNA, and tRNA is mRNA's messenger RNA carries the information. rRNA's ribosomal RNA and rRNA makes up ribosomes, which is the cellular structure that produces protein from the message that mRNA carries. And transfer RNA, tRNA, is the one that carries amino acid and the anticodon, which will match up to the mRNA. And so translation is mRNA to protein. And so what happens is this mRNA chain will go into the ribosome and the codons will match up to anticodons carried on the tRNA. This is the tRNA. And when they match together like complementary pairs, you can see when the complementary pairs match up, it releases the amino acid on top of the tRNA. And these amino acids bond to each other into a polypeptide, and the bond between them is called a peptide bond. And so codon matches up to anticodon. Anticodon releases amino acid, and amino acid forms polypeptide with peptide bond. And polypeptides fold up into proteins. And so how to find out which amino acids are released when we match up the codon and anticodon? We can use a codon chart. And as you can see, we have AUG, we have first letter A, second letter U, and G, and we have a met over here. Yeah. And so what is a protein? I think the question is, the protein is many amino acids, like thousands, millions of amino acids, billions even, billions of amino acids bonded together with peptide bonds into polypeptides that fold up into a structure, a macromolecule structure. And you can see it's like you have all these amino acids bonded up here, and you have the secondary structure, the tertiary structure, and we have this complex protein. And that's all. Thank you.